nucleus. So, this nucleus you can consider it as a master cell organelle, because it is controlling all the activities of the cell. It is controlling all other cell organelles. Is it clear? Right. And this nucleus, it contains genetic material. It contains genetic material. So, that is mainly DNA. It contains genetic material DNA. So, it is hereditary in function. So, it is hereditary in function. So, the nucleus contains genetic material, that is DNA. So, it is hereditary in function. Heredity means what? Transmission of characters from parents to children, or the transmission of characters from one generation to the next generation, that is called as heredity. So, here the genetic material is responsible for heredity. So, what is that genetic material? The substance which carries the information from one generation to next generation, that is called as a genetic material. Here we have various types of macromolecules like carbohydrate, proteins, fats and nucleic acids. Among these, the nucleic acids acts as a genetic material. So, what are those nucleic acids? The chemicals present inside the nucleus are called nucleic acids. Okay, if you observe the structure of this nucleus, this nucleus contains nuclear membranes, nucleoplasm, nucleolus and chromatin. So, nuclear membrane. So, these are the nuclear membranes. The thin layers surrounding the nucleus, they are called as nuclear membranes. So, inside this nucleus there is a fluid. This is called as a nucleoplasm. At one end of the nucleus there is a solid mass, and this solid mass is called as a nucleolus. And from this nucleolus, irregularly scattered genetic material form, and this is called as a chromatin. So, this is for your understanding. You have to remember, this is a structure of nucleus. So, the nucleus contains genetic material, that is DNA. So, it is hereditary in function. It is the main cell organelle. It controls all the activities of the cell. So, it is called as master cell organelle. If you observe the structure of the nucleus, it contains nuclear membranes and nucleoplasm. That means, the fluid matrix present inside the nucleus is called nucleoplasm. A nucleolus, a solid mass present at one end of the nucleus, that is called as nucleolus. So, this is nucleolus, and from this nucleolus, a irregularly scattered thread like structure formed, that is called as a chromatin. So, actually this chromatin is the genetic material. So, if you observe the composition of this chromatin, it is made up of DNA, and histone proteins. So, majorly it contains DNA. So, along with this, histone proteins are also present. Histone proteins means these are the basic proteins, just you remember that one. So, what is that chromatin? Chromatin acts as genetic material. What is the composition of chromatin? DNA and histone proteins. Histone protein means some basic protein, just to remember the terminology, that is enough. Okay? Yes. Right. So, this is the basic structure of nucleus. Next, let us see the nucleic acids. So, nucleic acids. What are nucleic acids? The chemicals present inside the nucleus. Okay. Chemicals present inside the nucleus. Chemicals in the nucleus, they are called nucleic acids. Okay. So, these nucleic acids they are made up of nucleotides, made up of nucleotides. So that means nucleotides are the monomers of the nucleic acids. Say many nucleotides, say for example, say this is nucleotide and this is nucleotide, 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 so on. So these nucleotides are linked by a phosphodiester bond. Phosphodiester bond. So this is nucleotide. So these nucleotides are linked by phosphodiester bond to form a chain-like structure that is called as polynucleotide. Polynucleotide. And all these polynucleotide forms the nucleic acids. Are you getting? So what are nucleic acids? These are the chemicals present inside the nucleus. Okay. And these nucleic acids are made up of nucleotides. Nucleotides. That nucleotide is a small unit. Is it clear? So many nucleotides link together by phosphodiester bond to form a chain-like structure that is called as polynucleotide. Polynucleotide. Now let us see the composition of the nucleotide. Composition of the nucleotide. And this nucleotide is made up of three components. So what are they? They are nitrogen bases, sugar and phosphate. Nitrogen base, sugar and phosphate. So the monomers of the nucleic acids are called nucleotides. So what are nucleotides? These are the monomers or small subunits which are made up of nitrogen bases, sugar and phosphate. All the three together called as a nucleotide. Nucleotide. And another terminology you remember, nucleoside. Nucleoside. So nucleoside is equals to nitrogen base plus sugar. Excluding the phosphate, then it is called nucleoside. So nitrogen base, sugar and phosphate is called nucleotide, only one letter difference. Okay, tide. But here nucleoside, nitrogen base plus sugar, excluding the phosphate, that is called as nucleoside. Are you getting? Right. So don't confuse here, slowly understand. So just I will revise once. Nucleic acids. What are nucleic acids? Chemicals present inside the nucleus. Okay. Nucleotides are the monomers of the nucleic acids. Okay. The nucleic acids are formed of nucleotides. Many nucleotides are linked by phosphodiester bond to form a polynucleotide, chain-like structure. Okay. So what is the composition of nucleotide? Nitrogen base, sugar and phosphate. Suppose if you exclude the phosphate, then it is called nucleoside. So nucleoside contains nitrogen base plus sugar. Up to here clear? Right. Next, there are two types of nucleic acids. Nucleic acids. Two types of nucleic acids. They are DNA and RNA. So DNA means deoxyribonucleic acid. RNA means ribonucleic acid. So deoxyribonucleic acid, ribonucleic acid. So this DNA is made up of deoxyribonucleotides. RNA is made up of ribonucleotides. Remember. So DNA made of deoxyribonucleotides. Deoxyribonucleotides. Whereas RNA is made up of ribonucleotides. Don't confuse here. Is it clear? Nucleotides are the monomers, smaller units of nucleic acids. Okay. There are two types of nucleic acids, DNA and RNA. DNA is made up of deoxyribonucleotides. RNA is made up of ribonucleotides. Ribonucleotides. Up to here clear na? Right. So let us see DNA. So there are two types of nucleic acids. They are DNA and RNA. DNA is made up of deoxyribonucleotides. RNA is made up of ribonucleotides. So let us see DNA. Let us see DNA. Here you have to remember some concepts here. Okay. So DNA means deoxyribonucleic acid. Deoxyribonucleic acid. It is made up of deoxyribonucleotides. Deoxyribonucleotides. It is made up of deoxyribonucleotides. So here nucleotide means what? Nitrogen base. Nucleotide means nitrogen base plus sugar plus phosphate. This is nucleotide. Got it? So let us see what are nitrogen bases. Nitrogen bases. Nitrogen bases. There are two types of nitrogen bases. Two types. Nitrogen bases, name itself indicating, they are basic in nature. They are alkaline in nature and they contain nitrogen. They contain nitrogen element, the nitrogen compound. So there are two types of nitrogen bases. They are purines and pyrimidines. Purines and pyrimidines. There are two types of nitrogen bases. They are purines and pyrimidines. Purines, again two types. So why they are called purines? They resemble a substance called puron. They resemble the structure of a substance called puron. That's why they are called purines. You don't need that one. Just it is for your understanding. And pyrimidines. Why they are called pyrimidines? They resemble a compound called pyron. That's why they are called as pyrimidines. Is it clear? Right. Just to understand that one. Purines are again two types. So they are purines are again two types. They are adenine, represented by capital A, and guanine. Guanine represented by capital G. Capital G. And these pyrimidines, the pyrimidines are three types. Pyrimidines are three types. So what are they? They are cytosine represented by capital C, thymine represented by capital T, and uracil represented by capital U. Capital U. First you remember we are discussing about the DNA, which is made up of deoxyribonucleotides. So nucleotide means three components are present: nitrogen base, sugar and phosphate. So we are discussing about the nitrogen bases. Nitrogen bases means these are the nitrogenous compounds. They are basic in nature. There are two types of nitrogen bases, purines and pyrimidines. Purines are two types. They are adenine and guanine. Pyrimidines are three types. They are cytosine, thymine and uracil. But the nitrogen bases present in the DNA are, so nitrogen bases present in DNA. Nitrogen bases present in DNA are adenine, guanine, cytosine and thymine. Thymine, you have to remember this one. The nitrogen bases present in the DNA are adenine, guanine, cytosine and thymine. Nitrogen bases present in the DNA are adenine, guanine, cytosine and thymine. So totally nitrogen bases are 5 types. So total nitrogen bases are 5. There are 2 types, purines and pyrimidines. Purines are adenine and guanine, pyrimidines are cytosine, thymine and uracil. Okay. So this adenine, guanine, cytosine, thymine, these 4 are present in DNA. Adenine, guanine, cytosine and uracil, they are present in RNA. That you have to differentiate. Okay. Right. So nitrogen bases present in the DNA are adenine, guanine, cytosine and thymine. Then nitrogen bases in RNA. Nitrogen bases in RNA, they are adenine, guanine, cytosine and uracil. Okay. So in DNA, adenine, guanine, cytosine and thymine. See, listen, these three, adenine, guanine, cytosine, these are common. Okay, these are common in both DNA and RNA. But uracil is present exclusively in RNA. Whereas thymine is present exclusively in DNA. This you have to remember. Is it clear? Right. So totally nitrogen bases are purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. The nitrogen bases present in the DNA, adenine, guanine, cytosine and thymine. The nitrogen bases present in RNA, adenine, guanine, cytosine and uracil. Okay, don't confuse this one. So simply you remember the nitrogen base present only in the DNA, thymine. The nitrogen base present only in the RNA, uracil. Or you will get the question here, which of the following will differentiates DNA from RNA? Thymine. Which of the following differentiates DNA from RNA? Means exclusively present in the DNA, that is thymine. Okay, and which one will differentiate RNA from DNA? Uracil. Up to here clear na? Yes. No confusion. Okay, right, done. Next, the sugar. So the sugar present in the nucleic acids is pentose sugar. It is pentose sugar. Pentose sugar means it is a 5-carbon sugar. 5-carbon sugar. Sugar in DNA, that is deoxyribose sugar. Whereas in RNA, the sugar in RNA, it is ribose sugar. So nucleotide contains nitrogen base and sugar. Okay, nitrogen bases are two types, purines and pyrimidines. Purines, adenine, guanine. Pyrimidines, cytosine, thymine and uracil. And then sugar present, pentose sugar. Sugar which is present in DNA, that is deoxyribose. In RNA, ribose. Now listen, it is for your understanding. Here ribose and deoxyribose both are pentose sugars. Pentose sugars means, first listen, don't take this one, it is for your understanding only. Okay. So suppose this is a 5-carbon sugar, say this is a 5-carbon sugar, pentose sugar. Okay, and this is the first carbon and second carbon, this is third carbon, this is a fourth carbon and this is a fifth carbon. This is the structure of ribose sugar. Got it? So first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Here HOH, this is for your understanding, don't take this one. HCH2OH. This is the actual structure of ribose sugar. Okay, this is a structure of ribose sugar. This is a pentose sugar, 5-carbon containing sugar. Got it? But in deoxyribose, in DNA, that is in deoxyribose, what happens means, at the second position, this oxygen atom is removed. At the second position. DD, removal of oxygen. Is it clear? That is the difference between ribose and deoxyribose. So ribose sugar is normal one. Deoxyribose, DNA, one oxygen atom is less at the second carbon atom, at the second position. Okay, that's why it is called deoxy. So there is a structural change. Is it clear? That's enough. And next, phosphate is common in both DNA and RNA. Is it clear? So this is the composition of DNA and RNA. Just you have to remember DNA, nitrogen bases, adenine, guanine, cytosine, thymine. RNA, adenine, guanine, cytosine and uracil. Sugar. So deoxyribose sugar in DNA, ribose sugar in RNA. This much is enough. Is it clear? Right. And then if you observe the structure of the DNA, this structure of DNA was proposed by the scientists called Watson and Crick. in the year in 1953. So, for that they got Nobel Prize in 1954. Okay. So, according to them, the DNA structure is double helical. Double helical. Double helical means, so like this, means it is made up of two strands, two polynucleotide strands. This is one polynucleotide, this is another polynucleotide. If you observe the composition, suppose say this is nitrogen base, adenine, sugar and phosphate. This forms one nucleotide. Got it? In a similar manner, thymine, sugar, phosphate. This is another nucleotide. This is for your understanding only. And these two are linked by phosphodiester bonds like this. Okay. This is nucleotide. This is nucleotide and this is phosphodiester bond. Phosphodiester bond. So, like that it forms many polynucleotides. So, like this. Okay. So, like this. So, in a similar manner here also in the other strand also. So, this is for your understanding. This is for your understanding. So, many nucleotides linked together by phosphodiester bond to form a polynucleotide. So, like that two polynucleotides are coiled together like this. Okay. This forms a double helical structure. This was proposed by Watson and Crick. For that they got the Nobel Prize. Is it clear? Right, that much enough. And here another point you have to remember, here in DNA complementary base pairing is there. Complementary base pairing means adenine pairs with thymine by 2 bonds, guanine pairs with cytosine by 3 bonds. This you have to remember. Okay. Always base pairing, this is called base pairing. Adenine pairs with thymine by 2 bonds, guanine pairs with cytosine by 3 bonds. A double bond T or T double bond A, G triple bond C or C triple bond G. This much you have, this one you have to remember. So, in a similar manner, in place of thymine in RNA you will get uracil. Okay. In place of thymine in RNA you will get uracil, that you have to remember. This is called complementary base pairing. So, RNA means ribonucleic acid. Ribonucleic acid, which is made up of ribonucleotides. Okay. So, the nitrogen bases present in the RNA are adenine, guanine, cytosine, uracil. Adenine, guanine, cytosine and uracil. Sugar is ribose sugar. Okay. Right, you know it very well. So, then RNA, it is single stranded means it is made up of single polynucleotide thread like structure, whereas DNA is double stranded. Okay. So, single stranded like this. Then 3 types of RNAs are there. The RNAs are 3 types. They are mRNA, rRNA and tRNA. mRNA, rRNA and tRNA. So, mRNA means messenger RNA, rRNA ribosomal RNA, tRNA it is transfer RNA. So, RNA it is single stranded one made up of adenine, guanine, cytosine and uracil nitrogen bases, ribose sugar and phosphate. There are 3 types of RNAs. They are mRNA, rRNA and tRNA. mRNA means messenger RNA, rRNA means ribosomal RNA, tRNA means transfer RNA. Now listen. Generally the genetic information, actually the DNA stores the genetic information. Okay, and from this DNA the genetic information will be carried in the form of mRNA, and from mRNA it will be decoded in the form of proteins. Okay, this is the sequence of transfer of genetic information. Got it? Generally, the genetic information is stored in the DNA, and this DNA, the genetic information present in the DNA will be transcribed to mRNA, and this process is called as transcription. Okay, from mRNA, the genetic information will be expressed in the form of final products called translation. Got it? Right. So here the genetic information present in the DNA will be expressed in the form of mRNA. This process is called as transcription. You have to remember the terminology, transcription. Okay. Now this information present in the mRNA will be finally expressed in the form of proteins, and this process is called translation. This entire process is called as central dogma of life. Okay, this sequence is common. This sequence is universal in all the organisms, plants, animals, human beings, or a microorganism. That's why it is called central dogma of life. This entire process, it is called central dogma of life. Now listen. So here mRNA means what? Messenger RNA, means it is carrying the message from DNA to the proteins. Messenger RNA, remember. Then rRNA means ribosomal RNA. It involves in the construction of the ribosomes. So what are ribosomes? Ribosomes are the cell organelles which involves in the protein synthesis process. So this rRNA is, the name itself indicating, it involves in it is useful for construction of the ribosomes. Next, tRNA, transfer RNA, means it is transferring the amino acids required for protein synthesis. It transfers the amino acids, for protein synthesis. That's why it is called as a transfer RNA. This you have to remember. Is it clear? So first you remember this sequence, DNA to mRNA, mRNA to proteins. These two terminologies you have to remember, transcription and translation. So now let us see this sequence, that is central dogma of life. So the genetic information will be carried in the form of mRNA. From mRNA, it will be expressed in the form of proteins. Okay. And this process, that means the conversion of DNA to mRNA, it is called transcription, and from mRNA to proteins, it is called translation. We know it very well. This entire process is called central dogma. Now just it is for your understanding, you remember this is a DNA. Say this is DNA, it is double stranded one. Okay, it is double stranded. In this DNA, the nitrogen bases are arranged like this. Complementary nitrogen bases. Okay, complementary means A pairs with thymine by 2 bonds, guanine pairs with cytosine by 3 bonds. Okay, this is called complementary base pairing. Now here one of the strand of the DNA, it acts as a base, okay, for the formation of RNA. Got it? Yes. So that means the complementary pairing will be formed. Suppose say A, G, C, T, C, T, G, C, A, G, like that, so on. Say this is a sequence, don't take this one, try to understand, that's enough. And complementary base pairing means here in RNA you will get, in place of A you will get T. T means what? RNA, in place of T you will get uracil. So in place of G you will get C. Okay, G, A, G, A, C, G, okay, U, C, C, like that, complementary base pairing. Got it? So like that the mRNA is formed here. So this is a mRNA is formed here. Got it? Okay. So now the sequences are here like this. So U, C, G, A, G, A, C, G, U, C, C, like that, so on. Okay, don't take this one, just understand. Here in mRNA, the genetic information will be stored in the form of a set of 3 nitrogen bases. In the set of 3 nitrogen bases. So these 3 nitrogen bases are called codons or genetic code, or it is also called as triplet code. Got it? So what is that genetic code or triplet code or codon? A set of 3 nitrogen bases. A set of 3 nitrogen bases forms a codon or triplet. Okay, just to remember. So the genetic information present in the DNA will be expressed in the form of mRNA. In mRNA, the genetic information will be stored in the form of a set of 3 nitrogen bases. This set of 3 nitrogen bases we called as codons or genetic code or triplet code. Okay. So here we have 4 nitrogen bases. Okay. They are arranged in the form of a set of 3. So how many combinations are possible? How many different combinations are possible? 4 power 3 means 64 codons are possible. Are you getting my point? Yes. So totally we have 4 nitrogen bases in the RNA: adenine, guanine, cytosine and uracil. So in that 4 nitrogen bases, 3 are arranged as a set: adenine, guanine, cytosine, uracil. Okay. Here this is one combination. AGU is one combination. Is it clear? GCU is another combination. Okay. And ACU is another combination. So like that, how many combinations are possible? How many different combinations possible? This is called probability. 4 power 3 means 64 codons are possible. Okay. Among these 64 codons, 61 codons are functional. These points you have to remember. 61 codons are functional, and 3 codons are non-functional. Means what? So this 61 codons will carry the information, and the remaining 3 codons does not carry any information. Okay. And these 3 codons, they are called as non-functional codons. They are called as stop codons. They are called as termination codons. Just you by heart this one. So these non-functional stop codons, termination codons are UAA, UAG and UGA. Just you by heart this one, that much is enough. Got it? So DNA, the information present in a DNA will be carried in the form of mRNA. Up to here clear now? Yes. In mRNA, the information may be stored in the form of a set of 3 nitrogen bases. This set you called as codons. This set is called as codons. Got it? So how many combinations are possible? 64 combinations are possible. Among these 64, 61 are functional. Okay, don't remember these 61. Just to remember the number. Total number of codons are 64. Among these ones, 61 are functional, 3 are non-functional. And these 3 non-functional codons, they are called stop codons. Means what? Suppose this is a mRNA. Suppose if you get UAA here, okay, UAA, as it does not carry any information, if it comes, the protein synthesis will be stopped here. Got it? The protein formation will be terminated here. That's why they are called as termination codons or stop codons. Just to remember this one. Okay. Right. So let us see. The next topic is, so before going to this genetic engineering, you have to remember some other terminologies like genome, chromosome and gene. Okay. So let us see genome. What is genome? Genome means a complete set of a complete set of genetic information of an organism. A complete set of genetic information of an organism is called as a genome. That means the haploid set of genetic information. A complete set means here you have to remember haploid set, that is called as genome. So, genome means what? A complete set of genetic information of an organism. Suppose if you consider any organism, if you take human being, the human being contains some genetic material that will be exist in the form of DNA. Is it clear? Yes. So, the entire DNA present in the human being, that you call as genome of human being. Okay. If you consider a dog, the genetic information, the genetic material present inside the dog, you call as the genome of that dog. That genome, that genetic material is responsible for its physical nature. That genome is responsible for its characteristic features. So, like that, each and every organism in the universe has its unique set of genetic information, unique set of genome, but the composition of all the genomes in the organism is same. That means the genome is made up of DNA only. What is the composition of DNA? DNA is made up of nitrogen base, sugar and phosphate. Is it clear? But where is the difference? Suppose if you consider the DNA, the structure of DNA is similar. It contains nitrogen base, sugar and phosphate. This composition is same in all the organisms, whether it is human being, plant, animal, whatever it may be. Then where is the difference? Here the sugar is same, that is deoxyribose sugar. Phosphate is also same. Okay, but nitrogen bases also same, adenine, guanine, cytosine and thymine. So, these are the nitrogen bases present in the DNA. Then where is the difference? Means difference in the quantity of DNA. Okay, some organism may have huge amount of DNA, some organisms, some microorganisms like bacteria, they have very small amount of the DNA. Okay, that means they differ in the quantity of the DNA. Quantity of DNA means quantity of nitrogen bases, and they differs in pattern of arrangement of nitrogen bases. Okay. So, this is the basic difference among the individuals. So, most of the organisms, they differs in the quantity of the DNA, and also they differs in the pattern of arrangement of or pattern of sequence of the nitrogen bases of the DNA. That makes the difference between one individual to other individual. That makes the difference of one genome to other genome. Is it clear?
Yes.
Right. Now, you remember. So, what is genome? A complete set of genetic information of an organism. That too, it is a haploid set. I will let you know what is haploid. First, you remember the terminology. Right. Next. Next, what is chromosomes? What are chromosomes? Chromosomes. So, chromosomes means the genome exists as specific structures. Genome exists as specific structures. That means they acquires, this entire genome acquires a specific shape, specific structure, that is called as chromosome. Say for example, if we consider a cell, this is the nucleus, inside the nucleus the chromatin is there, that chromatin is irregular in shape. At the time of cell division, it acquires a shape, specific shape like this. Okay, this shape we call it as a chromosome. Okay, the genetic information, the genetic material exists or appears as specific structures, acquires specific structures, and these structures are called as chromosomes. And the number of chromosome is specific to each of the individual, each of the species, you can say. Say for example, human beings, you consider human beings, all human beings in the universe have 46 number of chromosomes. Okay, that is its genome. So, that is the condition of chromosomes in the human beings. Is it clear? Right. So, like that the number of the chromosomes varies from one species to other species. Got it? And then what are genes? Then what are genes? Suppose say this is a chromosome, na? On these chromosomes a small functional units are present. They are arranged in the form of a linear structure. Okay? They are arranged in the order, and this one you call it as genes, means the functional unit of the DNA. Okay, functional unit of the chromosome you call it as a gene. So, what is gene? The functional unit of the chromosome or the functional unit of the DNA, a small section of the DNA within the chromosome which codes for protein. Okay. So, the functional small section of DNA, that is chromosome, which codes for a protein. Small functional section of the DNA or chromosome which codes for a specific protein, that is called as a gene. If you observe the genome size of humans, or you write down like this, chromosomes. If you observe the size of DNA in humans, size of DNA or genome in humans. So, it is 3 billion base pairs. It contains 3 billion base pairs. That means here the size of the DNA, size of the genome can be represented in the form of base pairs. How many number of base pairs are there? What is that base pair? That already told you, A double bond T, G triple bond C. Okay. So, this A double bond T represents one base pair, G triple bond C represents another base pair. Like that, how many number of base pairs are there? That represents the size of the DNA. So, you remember this one, the size of the DNA or genome on humans contains 3 billion base pairs of the DNA, 3 billion base pairs of nitrogen bases. That means 3 into 10 power 9 base pairs, you can say 3 into 10 power 9 base pairs. This is the size of a human DNA. Okay. Now, if we consider the chromosomes, in humans, there are 46 chromosomes. There are 46 chromosomes in humans. They exist as 23 pairs. So, the number of chromosomes in humans are 46, and these 46 chromosomes exist as 23 pairs. Okay. Another terminology you remember here, diploid and haploid. So, diploid condition, it can be represented by 2n, means if the chromosomes are paired up like this, then it is called as diploid condition. Okay. And here haploid, you remember terminology, haploid means it is represented by n. A single set of chromosome exists like this, then it is called haploid. Generally in the organism, the haploid condition occurs in the gametes. For example, gametes means sex cells like sperms and ovum. Sperms and ovum contains haploid number of chromosomes. Okay. Just you remember these two terminologies. Diploid means what? If the chromosomes are paired up, say this is one chromosome, this is another chromosome, this chromosome is from father, this chromosome is from mother. Okay. If these two chromosomes are paired up like this, got it? This condition is called as diploid condition. If they are individual separated, haploid condition. This haploid condition, individual condition exists only in the gametes. Gametes means sex cells. Okay. Say this is sperm. So, in sperm 23 chromosomes are there, they are not paired up. This is ovum from mother. So, here also 23 chromosomes are there, but they are not exist as pairs. Is it clear? Individuals. So, this individual condition we call as the haploid condition. If they get paired up, then it is called diploid condition. Got it?
Yes.
Right. You have to remember this terminology, diploid and haploid. Diploid means chromosomes are paired up. Haploid means chromosomes are separated. Okay, this haploid condition exists only in the gametes, that is in the sex cells, reproductive cells, okay, during the formation of sex cells. Is it clear? And this diploid condition exists in all the vegetative parts of our body. If you take a skin cell, if you observe the chromosomes, they exist in diploid condition. If you take a hair follicle, so if you observe the chromosomes, then they exist as diploid condition. Is it clear? Except in the gametes. Got it? Right. Now, you remember these two terminologies, diploid and haploid. Let us come to here. In humans, there are 46 number of chromosomes. These 46 chromosomes exist as 23 pairs. So, this 23 pairs means 23 from father, 23 from father and 23 from mother. Okay. So, that is a sperm and this is a ovum, right. And these 46 chromosomes, there are two types. These are of two types. So, one is autosomes, or they are also called as vegetative chromosomes. And another category is allosomes, or they are also called as sex chromosomes. Okay, right. So, these 46 chromosomes exist as two types. They can be divided into two types. One is autosomes or vegetative chromosomes. There are 44 autosomes. Allosomes are sex chromosomes. There are two chromosomes. And these allosomes are sex chromosomes. They are of two types again. They are X type and Y type. So, totally 46 chromosomes are present. They exist as 23 pairs. And these 46 chromosomes can be classified into two categories. One is autosome and another one is allosome. These autosomes are called vegetative chromosomes. They control the vegetative characters. And allosomes are sex chromosomes, they control the sexual characters, right. And here these 44 autosomes are similar in both males and females, whereas the allosomes, the sex chromosomes, are of two different types, X type and Y type. Okay. They makes the characteristic features of both males and females. If you observe the males, if you observe the composition of or combination of chromosomes in males, there are 44 autosomes plus XY allosomes, XY allosome. You remember this one. 44 autosomes and XY allosome. Whereas in females, the combination is 44 autosomes and XX chromosomes, Okay. This you have to remember. The males have 44 autosomes and XY allosomes, whereas females have 44 autosomes and XX allosomes. Here you remember these 44 autosomes are similar in both males and females, and XY combination makes the masculine characters, XX combination will make the feminine characters. Is it clear?
Yes.
And here also one thing you remember, X chromosome is present in both males and females. So, specifically Y chromosome is present in males. So, that Y chromosome will decide the sex of the baby, that you have to remember. Is it clear?
Yes.
The Y chromosome during fertilization will decide the sex of the baby. Is it clear? I will let you know that one. Up to here clear now?
Yes.
Huh? Right. So, 46 chromosomes, autosomes, allosomes. Males have 44 plus XY, females have 44 plus XX. Generally, this is the normal number of chromosomes and composition of the chromosome, combination of chromosome. If there is any alteration occurs in the number of chromosomes or in the structure of the chromosomes or in the pattern of arrangement of the chromosomes, then it leads to genetic abnormalities, then it leads to chromosomal disorders or genetic abnormalities, you can say, that cannot be rectified by any doctor, any scientist in the world. Is it clear? These type of abnormalities we call as congenital abnormalities. Congenital abnormality means these abnormalities occurs by birth, from birth you can say. Is it clear? So, these are called congenital abnormalities.